Hi everyone, my name's Brierley and I'm from the University of Salford studying a PhD, and today I'm going to talk to you about anaplasma phagocytophilum sequencing technology to explore its diversity in epidemiology. To give you a bit of context, this is the Rickettsialis order, and the anaplasma genus is thought to have evolved around 500 to 600 million years ago at the end of the Neoproterozoic. One of its constituent species is anaplasma phagocytophilum, which are referred to as AP, because it's a lot easier to say. And this is a globally distributed tick-borne bacterial parasite. It invades the granulocytes, particularly the neutrophils, but sometimes the eosinophils. And it's the causative agent of granulocytic anaplasmosis, which is an immunosuppressive febrile illness. In Europe, it's a major contributor to lameness in UK ruminants and just generally sheep across the UK. But in North America, we find that it's actually more of a human infection. So you see a lot of human infections. I think up to around 15,000 per year are being reported. And as such, it's a major economic burden that impacts both animal welfare and human health in North America. So we've known about AP for around 100 years now. It was first discovered in Scottish sheep, and we've got some control strategies to sort of mitigate its effects. So firstly, we can provide the animals with doxycycline, which is a pretty effective antibiotic at getting rid of the disease. We've also got some crude vaccines. These are not so effective due to AP being able to modify its major surface antigens. We can then also spray acaricides, which will just reduce the number of ticks biting your animals. And we can also avoid tick-infested pastures. And then generally, farmers will rear the lambs in protected pastures for the first 6 weeks of life so that they can build up their innate immunity, so that when they do encounter the disease, they're more capable of fighting it off. And then lastly, it's very important that you choose the correct breeds. Breed choice is really important. It's just some breeds are far more naturally resistant than others. So in Europe, we know that Ixodes ricinus is the primary tick vector, but we also know that Ixodes frontalis and Ixodes triangulariceps are very competent vectors too. We know AP is maintained by a range of different wildlife species. These include deer, rodents, birds, and even reptiles now. But we don't know the roles these species of tick play and wild animals play in maintaining the important strains of APs to veterinary strains. There's a number of reasons for this. Firstly, AP is an obligate intracellular bacterium, so it's very difficult to isolate and grow. Secondly, there's a limited amount of data, which is primarily its point one. So this is generally limited data in terms of whole genomes. We do work with a lot of single gene data. And thirdly, there's a lesser research drive for this particular tick-borne parasite. You usually see other things like Lyme borreliosis taking a lot more of the attention. So in Europe, we generally base a lot of epidemiology on AP on the GROEL gene and the different haplotypes that are circulating around Europe. So this figure here is a really good illustration of that. You can see the 4 different ecotypes kind of spread out with the different mutations. So each blue dot represents a single base mutation. So you can see that group 1, so ecotype 1, is the largest group. And this is where we tend to find most of the veterinary infections. Group 2 we generally find in roe deer, but to an extent also in domesticated livestock. Group 3, or ecotype 3, we tend to find only in rodents and ticks. And then 4 we've only found in birds, and it seems to be quite a rare one. Which leads me on to the hypothesis of this project. So we believe that ecotype 1 is a generalist ecotype vectored by Ixodes ricinus and is responsible for the vast majority of veterinary infections. We think ecotype 2 is a roe deer specialist, also vectored by Ixodes ricinus. We think ecotype 3 is a rodent specialist, which is vectored this time by Ixodes triangulariceps. And ecotype 4 is a bird specialist, vectored by Ixodes frontalis. So to sort of try and begin to answer some questions about epidemiology for AP, we generated a 3-phase project. So the first phase was to sequence the first UK isolate, so it's never actually been sequenced before from the UK. The second phase was then to develop a protocol that would allow us to sequence AP without having to culture it in tick cell lines. And then the third phase was just to then make sense of all the data that we're generating. Phase one, yeah. This basically involved working with the University of Liverpool and their tick cell biobank, which have been collecting UK isolates of AP for around 50 years, I believe. And they grew up these isolates, provided it to us for sequencing, assembly, and annotation, which was in part done by Microbes NG. And then we used this data to kickstart phase two of the project. So these are the samples that we were working with, so 7 in total, 5 from sheep, 1 from a feral goat, and 1 from a cow. And these are grown up in ISE6 or IDE8 tick cell lines, which are derived from Ixodes scapularis, harvested at maximum bacteremia, and then the DNA was extracted. So the raw Illumina data was generated by Microbes NG, and we did the ONT stuff at the University of Salford. We got the raw data, put it through a quick quality filter, removed the adapters, and then we had to map it on the Kraken 2 standard database because we're sequencing both the tick cells and the pathogen inside them. So we need to separate that out for the assembly process. And then we put them into Unicycler, which is just a hybrid assembly software, and annotated them with the RAS toolkit. And so this is what an AP genome looks like. As you can see, it's a complete assembly for the Harris strain, which came from Scottish sheep. And the genome length is just over 1.5 megabases, a low GC content of 41.84%, which is typical for Rickettsia bacterium. We believe there's 1,645 coding genes, 704 of which we couldn't assign a function. And if you look at the subsystem analysis there, you can see the vast majority of genes are involved with metabolism and protein processing, which is also very typical of this kind of bacterium. We can then make some sense out of the data by putting it in the context of all the whole genome data available to date. So currently there's 33 genomes available on NCBI. We've got 2 here from collaborators in Amsterdam, and then we've got the 7 that we've sequenced from the UK. And you can see this on this phylogenetic tree, which is generated from 185,000 amino acids alignment, that our strains sit within European ecotype 1, like the vast majority of data that's currently been sequenced. I think what's really interesting about this tree, though, is that the sheep and the cow strains that have come from the UK and France do cluster into sort of an Anglo-French clade, and they are separated. So it suggests sort of a geographic and host basis for evolution. I think it's also interesting that you can see that the European ecotype 1 is even more diverse than all of the strains in North America combined. And we believe that the North American strains have actually recently been transferred over in the past few thousand years to that part of the world, and that it's the founder effects are really responsible for this low amount of diversity and why we see so many human infections in North America. Moving on, so phase 2 of the projects, this was to basically develop a protocol that would allow us to skip the whole cultivation stage, which is very lengthy, time and expensive process to get through. So this involved the extraction and quantification of AP DNA from blood and spleen samples, the evaluation of some enrichment techniques, because it's not very easy to sequence. It's basically metagenomic sequencing, and then the attempt of sequencing, assembling, and annotating these genomes that we're getting from the data. So we looked at several different species. Firstly, we looked at Cumbrian sheep, which I went and got from Denny's abattoir. Not the nicest thing to do, but it was worthwhile. We found 8 of these were infected out of the 80, so a 10% infection rate. CTs range from 25.2 to 30. We then also worked with Forestry England, so in Grisdale Forest, very close proximity to where the sheep would have been taken from. And they provided us with 23 red deer and 50 roe deer. We found that the red deer had an infection rate of 52%, so a lot higher, with CTs ranging from 23.5 to 27.7. And then with the roe deer, we found an infection rate, really high infection rate of 91%, so pretty much all of them were infected. And they also had the highest level of infections, so CTs as low as 18.8. We then also thought, for the sake of the ecotypes, we'd try and get some ecotype 3 samples. So we went to Kielder Forest to trap some more small mammals. We were looking for field voles. We only got 3 of them. And this was when the weather was really hot, so they just weren't coming out. But we did get some bycatch we've got some common shrews, and we did find that they had an infection rate of 31%. CTs ranged from 23.5 to 26.5. Then tried to benchmark sequencing efficiency. So I took 2 samples, CTs 21 and 20 from roe deer, and just put them into a MySeq V2 300 cycle kit, generated some data, then mapped it against the Kraken standard database to see how many reads were phagocytophilum group. And basically, there was nothing. There was about 3 reads. So it was completely useless for any kind of whole genome assembly, and you couldn't even look at any genes from this kind of data, which is sort of what we expected. And I think it's worth noting that the CT values we are generating are based on a multi-gene assay, so the MSP2 gene. There's about 100 copies of them in the genome, so it does artificially inflate that CT. So then we had to look for some sort of enrichment techniques to increase that number of reads that we're getting in the data. So the first thing that we looked at doing was using biotinylated baits. So the way these baits work is we concatenated the 7 genomes that we got from phase 1 of the project, and we designed baits that were complementary to basically every point of this genome. So there's 125,000 of them. And these baits are then, they have a biotin molecule attached to them. And during the library prep protocol, you put them in for hybridization. They'll bind to everything that's complementary, so you target epDNA, and then you put your magnetic beads laced with streptavidin in, and these will pull out all your target DNA with your baits, and then you do a PCR, bring it all back up, and then sequence. We did this for 8 different samples, 2 sheep, 2 red deer, and 4 roe deer, with a range of CTs just to see how CT would impact this. Then we also did much simpler in silico enrichment, which is Oxford Nanopore's adaptive sampling. And the way this works is you provide a reference sequence that the system can align to. So we provide the Harris genome. And when a molecule is being sequenced through the pore, after around 500 base pairs, it will try and align to that reference sequence that you provide. If it aligns, then it will accept the molecule and finish sequencing the whole molecule. If it doesn't align, it will reverse the charge, which will eject the molecule, allowing a new molecule to come in, basically enriching for the samples you want. And we did this again for the same 8 samples with a range of CTs, just to see how they compare these 2 different techniques. So to sum up, adaptive sampling, not very good. The baits, pretty good if you look at the high infection intensity samples. So yeah, with adaptive sampling, we got about 20-fold increase at best, where with the baits, we got at best a 27,800-fold increase in the number of reads, which is good, but 2.78% is still not a lot in terms of sequencing efficiency. So we're also looking at some extra upstream enrichment techniques. So a bit more old school, looking at the Molzyme Ultra Deep Microbiome Prep Kit, which is a differential lysis kit. So you lyse your host cells first, digest your host DNA, then lyse your microbial DNA, and then collect that DNA. And we're also looking at the NEB Next Microbiome Enrichment Kit, which is completely different, exploits differences between prokaryotic and eukaryotic DNA. So eukaryotic DNA has got a lot of methylation sites. The proteins on the beads will then bind these methylation sites, pulling it out of the solution, leaving behind your prokaryotic DNA with an enrichment. So to conclude, complete phase 1 of the project. We've sequenced and assembled the first 7 AP strains from the UK. And we made significant progress in phase 2. So we have got it to a point where we could start sequencing genomes, but for the sake of efficiency, we want to improve them enrichments a little bit more. And even though I've not mentioned phase 3, we have made a lot of progress on this because we've developed a lot of the bioinformatic pipelines that we need to make sense of all this data. So I'd also like to acknowledge that this is funded by the Perry Foundation, UKRI. It's a collaboration between the University of Liverpool, University of Salford and the National Institute for Public Health and the Environment in the Netherlands. Thanks for listening.